Hi, this is Amy Proal with the Polybio podcast. And my guest today is Dr. Lena Pernas. She's a research group leader at the Max Planck Institute for Biology of the Aging. And her lab studies Toxoplasma gondii, a parasite that causes a chronic infection in a large segment of the human population and has been implicated in a range of neuropsychiatric symptoms. Lena is currently studying if chronic toxoplasma infections are reactivated in patients with COVID-19 or LongCovid. And she's also an expert on the molecular mechanisms by which toxoplasma modulates the metabolism of the cells that it infects and how mitochondria function as part of the innate immune response that fights back against the parasite. And with that, welcome, Lena.
Thank you so much, Amy, for the invite. It's really exciting to be here and have the chance to talk to you.
Absolutely. Lena, all right. So when I talk about Toxoplasma gondii, can you tell people more about Toxoplasma? What is it? It's a parasite. Who does it infect? How do people get it?
Absolutely. So from those of you listening, if you have heard of Toxoplasma gondii, you've probably heard of it in the same sentence as cats or poop. And that's because cats are the definitive host. And why that's relevant to us is that it means that a cat can shed billions of parasites in its feces. I think this tends to terrify people and think they need to get rid of their cats, but it takes about 10 days for the parasites that are in the feces to become infective. So as long as you clean your cat litter, you're fine. We normally get it from foods that have been contaminated with cat feces or from animals that we consume, or the meat that we consume that's undercooked that's been previously infected with toxoplasma. So that's how we normally get it. So you ask, who does toxo infect? Well, toxo is quite an incredible parasite because it's a single-celled eukaryotic organism, but it really has an unparalleled ability to affect any mammalian host. So from humans to, if you PubMed or Google's toxo mammalian host, you'll find the most random animals in which the parasite has actually been found in, from black-footed penguins to kangaroos. So it's a very successful parasite. It can infect, as I mentioned, probably any mammal. Humans, it's quite ubiquitous, so it's estimated to infect about 1/3 of the human population. Now, this can vary dramatically with country and with age demographic. So, for example, in Germany, the country I live in now, there was a study done about 5 years ago where they profiled the serology. So this enables them to look at whether or not there's an antibody signature to the parasite. So have people been previously infected? And they found that individuals of the age of 20, about 20% of those were infected. But as you get older, you have a much higher chance of being positive. So they found that about 60% of the population over 60 was infected with toxin. I mean, there's some regions of Brazil, for example, where more than 80% of the population is considered is shown to be seropositive. So wide distribution, very successful parasites. 1 in 3 of the people that you know are probably infected.
Wow. I don't think many people know that, I'll be honest with you. Now, when you say people are infected, obviously then the parasite can persist in people in a latent type form then, where it doesn't drive overt symptoms, because obviously 1 in 3 people I know don't have chronic symptoms. Is that correct then?
Yeah, no, I'm very glad that you brought that up. So the parasite can establish either an acute infection or a chronic infection. Acute is really considered to be the first 6 months. Now, for most immunocompetent individuals, so folks who have a strong immune system, they're likely not going to show overt symptoms. It has been shown to cause certain eye diseases, swelling of lymph nodes, and sort of flu-like symptoms, but it's generally thought to be asymptomatic, even in the chronic phase in immunocompetent individuals. It can then establish a chronic phase. So it's thought that once you're infected with toxo, you're infected for the rest of your life. And the reason you don't hear 1 in 3 people of your immediate contacts complaining about toxo-related symptoms is because it's true. It's been thought that once you're infected chronically, which just means you've been infected for more than 6 months, the parasite is in a quiescent, dormant phase where it's not actively disturbing your immune system or the infected individual in any way. But there's more and more reports emerging that while we think the infection is asymptomatic, it's really driving or associated with certain symptoms. So our own work from a while back had shown that there's certain cytokine signatures, so immune modulators in your blood that are associated with chronic infections in a population or patients from America, also patients from Colombia that we had looked at. So I think this concept of really an asymptomatic chronic infection, we're probably going to learn that in fact there are changes, just changes that we're not aware of that the parasite is driving.
Yeah, that sounds pretty similar to Epstein-Barr virus or some of the herpes viruses,
Exactly.
where more than, in that case, more than 90% of the population harbors one or more herpes viruses, and you have people who are healthy, and then yet you have people who have some issues or symptoms that begin to be tied to that. So there's kind of a middle ground of what the pathogen may be doing in some people where it's maybe active to a certain extent, I guess.
Yeah, absolutely.
Yeah.
I think it raises such an interesting question of all of these pathogens that are able to establish chronic infection. So it's sort of taken for granted that most of us have CMV or most of us have Epstein-Barr virus, and we think that it's asymptomatic, but in the end, it is an infection. And I can't remember the author now, but it's I think some studies have shown that the majority of IgGs that are identifiable in age populations are towards CMV. So either the virus is breaking out of a chronic phase and sort of replicating, but then is checked by the immune system. But you can imagine that this might have consequences on the ability of your immune system to prepare or to deal with secondary infections, for example, or what happens when you have stress in the form of a second infection. So what happens now when you're harboring two infections? How does your immune system deal with that? So I think more and more, not only in the case of toxo, but all these viral infections you mentioned, will appreciate that these latent infections probably have profound ways in which they're affecting our own physiology or ability to respond to secondary infections.
Absolutely. Actually, there's a Peter Broden paper that shows how cytomegalovirus modulates the immune response. He took twins who were discordant. One twin had the virus and one twin didn't. And he just shows how the immune parameters in the twin that are infected change. And it really shows you that just harboring that virus inevitably would change the way you would respond to a subsequent infection or deal with another pathogen. I think it's a good example. That then brings me into the case when you're mentioning multiple infections and potentially, in simple terms, reactivation of Toxoplasma or more activity of the parasite in some people. Are you studying that in patients with COVID then, COVID-19? And is that occurring?
Yeah, so we're very interested in asking. Well, we have a general interest in chronic infections, and since we're working on tox, of course, this is the first one that we'd like to look at. But the questions that we're interested in is really, are chronic infections associated with more severe COVID progression? And the reason we think that is that there's likely two possible ways that we can envision this occurring. The first is that if you have a chronic infection, from, for example, the study that you just mentioned with regards to CMV, but there is other work with looking at toxo and CMV and showing that these chronic infections do or are associated with changes in your peripheral blood cytokines. So again, the proteins that modulate the responses of your immune cells. So that means if a patient is infected with toxo chronically, there's a likelihood that they will have a different immune response to uninfected individuals that are getting infected. Now, this, of course, could have a beneficial outcome or a detrimental outcome if now the patient has a skewed immune response to SARS-CoV-2. So I think that's one way we can imagine chronic infections altering the ability of an individual to respond to coronavirus. A second possibility or a second way we imagine chronic infections interfacing with a secondary infection is simply through increased pathogen replication. So for a long time, I think the effects of Toxoplasma, for example, were not appreciated. But then during the AIDS epidemic, individuals that had latent toxo now started having all these severe symptoms because the parasite can actually live quietly in eye or brain tissue, for example. Now these patients that had an immunocompromised system started getting these brain lesions that were indicative, of course, of toxo reactivation and proliferation, so tissue damage. This is, I think, a possibility in a secondary infection because this poses a stress that might lead to the reactivation of a chronic infection like toxo. And if Toxoplasma begins proliferating, perhaps unchecked because maybe the immune system is more focused on dealing with coronavirus, for example, you could imagine that this might have negative consequences on the tissue in which the parasite is proliferating. So right now we're working with Dr. Jose Montoya at the Remington Toxoserology Laboratory to try and investigate whether patients that have been infected with COVID, if there's an increased probability that they have a chronic infection like Toxoplasma, but we're also checking CMV and Epstein-Barr virus, as you'd mentioned, and a couple others. And since we also have patients from these samples at different time points after diagnosis, we're asking whether or not the parasite is sort of reactivated from its chronic phase to begin proliferating again. And there's ways we can, or different ways we can do that. For example, we can look for antibody signatures, whether or not they have increased levels of IgGs, for example, that indicate that the parasite has reactivated and the immune response to it has been mounted. We can also look for levels of toxo-DNA, for example, in plasma from patients. So hopefully we'll have answers in the coming years to this.
That's awesome. That's two incredible studies that need to be done. I think, and you're doing that in acute COVID. Are you also going to try to do that when patients with long COVID? I mean, I'm sure you know that many people are developing a broad range of chronic symptoms after COVID. Do you think that toxo may remain activated or may have infected brain tissue or other tissue in a more detrimental fashion in a manner that then can persist over time and then actually keep certain chronic symptoms going in patients?
Yeah, I think that's a really interesting possibility. I mean, so to short answer your question, I wish we had access to long COVID patients. We don't yet, but this is, of course, if you know of any collaborators you can put us in touch with, we would love to do this. But as you mentioned, it's possible that during acute COVID infection, that the parasite reactivates and then causes damage that might lead to symptoms that are associated with long COVID, or that the parasite itself is still present at basal levels. I think it could be very interesting to look at different markers of chronic infections, whether that be at the level of antibodies or microbial DNA in these patients, especially if you have access to sort of kinetic analysis or patients over time following diagnosis. I think this would be super interesting to look at.
Absolutely. I'm going to see if I can find you a cohort of long COVID patients. Yeah. No, because yes, I really think about that because, and it doesn't have to be toxo, but I always think about it. There's a lot of patients reporting neuropsychiatric symptoms after COVID, long-term neuropsychiatric and cognitive symptoms. Now, Toxoplasma, what kind of symptoms does it drive? So let's say it does become more active in simple terms. What would you expect to see just in terms of a patient who's suffering from this? Neuropsychiatric stuff or just flu-like symptoms too? What kind of symptoms?
I think it's an interesting and tricky question to address because it all depends on how you reacted the first time you were infected. So were you able to squash the infection pretty quickly such that you only have chronic infection set up in one tissue, or did it take you a while? So now you have chronic infection set up in different in cardiac tissue as well as eye tissue as well as brain tissue. So the symptoms that have been associated with reactivation can be quite severe when a neurological infection has been established. So for example, encephalitis is a big cause of reactivation of toxo infection in the brain. There's been, in healthy patients, there's been a lot of interesting reports about the way that Toxoplasma chronic infection affects an individual's behavior, from making, I'll address sort of the effects on the male population that have been identified, but inducing decreased activity, I guess, has been one. I recently read some reports that it makes, that patients that are chronically infected are more likely to start their own business or exhibit entrepreneurial activity. And apparently this is a global trend. I don't know the details in these papers, so I can't vouch for them, but there are interesting correlations in mammals. For example, in rats that are infected that are normally averse to cat urine or afraid of cat urine, it's been shown that this aversion is lost and the rats instead become attracted to cat urine. So there's all these interesting bits of data suggesting that toxo can have a dramatic effect on behavior and on different aspects of different organ functions. But with regards to the neurobehavior, it's really hard to tease apart what's actually causal, because this is all in patients. And so unless we're able to do this in a more experimentally controlled manner, they're just associations at best.
That's fair. It is really interesting, though, just the general trend of pathogens being able to modulate the behavior of the host that they infect, whether it's rabies or other pathogens like that. I don't think people take that into consideration sometimes, that, what we see a lot of research on behavioral changes or cognitive symptoms or neuropsychiatric symptoms in a lot of patients. And I do think that these latent infections need to just be considered or put on the table as some of the contributors to that across the board in patients with a range of diagnoses. I don't know, seeing as so many people can harbor them, right?
Yeah, no, I agree completely. And it's known that they harbor them. So this, I mean,
right.
There's people who you might have known who have gotten shingles after reactivation of, right. So it's possible. I'm also surprised. Well, when we became interested in this topic, we were very surprised that not much has been done on this. And I guess I can understand if you're coming in with a fulminant, let's say COVID-19 symptoms, perhaps toxo is not the first thing one wants to look at, but I think it's something that can be considered in terms of what might be exacerbating the symptoms in a patient who has acute COVID disease. So
yeah, absolutely. Now, can a doctor, if they are considering toxoplasma, how much can you identify it clinically? So are there tests, let's say a patient wants to know they have COVID, could toxo be involved in my symptoms? Can their clinician assess that? Or is that something that has to still be done more in a research setting now?
I think it would be feasible for a clinician to assess, given the presence of multiple samples from that patient. So if you only have one sample from a patient, then you only have one snapshot of your toxo response profile and antibodies. So IgMs, which are sort of your first antibody responders to infection, or IgGs, you won't know whether or not this is a secondary infection, for example, that's been reactivated, or the first time that maybe you were co-infected with toxo when you had COVID. So I think if a patient does, or if a clinician has access to multiple samples of an infected patient, and you can see clear changes and perhaps symptoms that have emerged, and some that might be more aligned with the profile you'd expect of toxoplasmosis, then I think it would be very interesting to ask what other diseases might have been awakened in this patient. If not toxo, perhaps CMD, perhaps EBV. I think in the clinic nearest the Uni Clinic, I think they have been testing for at least CMD in patients that are infected. So I hope that more and more this might inform what could be, why certain cases of the disease might result in such severe symptoms versus others.
Yes, definitely. And I agree, there have been clear reports of Epstein-Barr virus reactivation, or varicella, the chickenpox virus, or those viruses. So it would be hard to imagine if toxoplasma was not reactivating in some people too.
And the cases that have been reported have been very severe disease. So of course, these are one-off instances that have been reported, but I think the data or the evidence is tantalizing, and I hope this opens up more research into this area.
Definitely. I guess thinking of that, can you treat toxoplasma?
Yes, you can treat the acute phase, but it is not possible to treat the chronic phase. So there are drugs that clinicians can prescribe that can treat the acute phase, so the actively replicating phase. But once you're infected for life, and we do not have treatments for the chronic phase of the parasite.
Seems like that's an area that needs more development too. Although I suppose if you strengthen the immune system a little bit so that it itself could control the toxoplasma, again, that wouldn't hurt. But overall, that's interesting. So no necessarily existing chronic toxo therapeutics. Okay.
Exactly.
Okay. Really interesting. Okay. So, you've studied a lot about toxoplasma besides just what, when and how it might be able to reactivate, or that's your ongoing work. You've actually also studied how toxoplasma can, in simple terms, modulate the metabolism, or even hijack, in simple terms, the metabolism, the mitochondria of the cells it infects. And then in a really key, how mitochondria fight back as part of the immune system to that hijacking. Can you explain that more? Because it's fascinating, and it really speaks to a lot of what these pathogens do, because a lot of teams will study the presence or absence of a pathogen, but it's cool that you've gone beyond that with toxoplasma to study its activity.
Yeah, so I'm glad this is also something you mentioned. This is actually the major focus of my lab's work, is really trying to understand how toxo interfaces with host organelles and how host organelles respond in kind. I think the prevailing perception has been that microbes infect, and microbes are able to manipulate or exploit host organelles for whatever they need. And host organelles like mitochondria, which are our powerhouses or that make the energy required for life, are exploited by the pathogen. And I think there's been several revolutions in our understanding of mitochondria, and that we now, I think, perceive them as very, at least from a cell biology perspective, that these are very dynamic organelles that can regulate a diverse set of cellular processes. But yet, when you put the lens of infection on, they're still considered static bystanders. So a while ago, we had seen these interesting changes that occurred during infection with toxo, in which the mitochondria, well, I should say this was actually described about 50 years ago, that mitochondria traffic to the site of where the parasite is. But a while back when I started my postdoc, I'd noticed that the mitochondria actually looked a bit different. So their shape and the behavior was changed, which led me to ask, is it possible that these organelles that we for so long have considered static bystanders during infection are actually driving a host response against the pathogen? And one way we imagine this could happen is through a sort of nutrient competition. That's because mitochondria, which I told you are our powerhouses, also need nutrients and metabolites from the cell for their own biogenesis or generation, also to make our energy units, so ATP. Similarly, intracellular microbes also need this energy. And mitochondria are sort of derived from microbes. So we thought that perhaps there might be some sort of metabolic defense that the mitochondria were driving. What we found was that the changes in the shape that we found in mitochondria really underlied a metabolic change in the organelles, in which they were able to increase their ability to take up certain lipid species, which limited what the parasite had access to and restricted parasite growth. So mitochondria are acting as nutrient competitors to the parasite and limiting their ability to grow.
Wow. Yeah, it's fascinating because, yes, exactly. Most people still picture mitochondria as if they were in their biology textbook from high school, this static organelle, this little thing that just doesn't do anything. But in that sense, it's part of host defense. I've heard people say mitochondria are part of the innate immune response. Then you agree?
Absolutely. And I think, well, the work from, I think, 4 groups really put this on the map around 2005. A protein that is now called mitochondrial antiviral signal protein, MAVS, was found on the outer mitochondrial membrane. And this protein is really important for mounting a response to virus. So that really put them on the map as parts, or at least key signalers in innate immune responses. But whether or not they could sort of actively respond or autonomously respond to pathogens in a different way was something that I think is not yet really appreciated, and something that we're trying to find different ways in which mitochondria are defensive organelles rather than just static bystanders. And it's well known that several microbes can actually target mitochondria. I think if we ask the question of, well, why is this? Why have so many pathogens evolutionarily converged on targeting mitochondria? I think that's probably because mitochondria are so key in defending cells against microbes. And I hope we will find more ways in which mitochondria are able to do so.
Yes, that makes a lot of sense. So the mitochondria fight the pathogens, so the pathogens evolve to better fight back against the mitochondrial activity, and they're sort of evolving in concert there. That's really interesting. You know, I've seen teams when mitochondria, as part of their defense, and I think you have shown this in some of your papers, they fractionate, right? They break up. Is that considered sort of a normal part of their defense processes?
So it's interesting. I think it's not the mitochondria, as you mentioned, the breakup, we call that fission. So they're able to not only fuse, come together and sort of elongate, but they're able to divide. And it's not, I don't think we can really categorize any shape of mitochondria as always beneficial in an infection context. It depends on the need of the pathogen. It depends on where the pathogen resides in the cell. So for example, we think that the ability of mitochondria to take up fatty acids defends against Toxoplasma. But other researchers, Barbara Burley at the Public School of Health at Harvard, has found that for Chagas disease, or the parasite that causes Chagas disease, Trypanosoma cruzi, beta oxidation actually seems, or the ability to take up fatty acids actually seems to be good for that parasite. So I think it depends on the niche of the parasite or microbe, what that microbe needs. And in some cases, the fission of mitochondria may induce a response or may lead to, for example, increased glucose consumption by the rest of the cell because now mitochondria aren't working. Well, you can imagine some bacteria really need glucose. So now if the cell becomes more efficient at processing glucose, maybe there's less available for the bacteria. So I think the shapes give us an idea of whether mitochondria are responding to the pathogen and are a very clear way to identify, hey, mitochondrial behavior is changing. Maybe there's something interesting in the way that they're changing. But whether or not we can always say, this is good in the context of infection or bad, I wish it was that simple, but I don't think it is.
I get you. In other words, you could say something's going on. In other words, if you see fission, it likely means the mitochondria is responding to a pathogen, something's happening, it's trying to act in a defensive way. But of course, there can be some variability in how that plays out for its own ability to function normally during that time versus how it's targeting the pathogen by doing that. Okay, it's really...
And I'm very mitocentric, but of course that could also reflect the pathogen actually targeting mitochondria and releasing an effector protein that can maybe target and inhibit a certain function in the mitochondria.
Yeah, definitely. Interesting. When it comes to the fact that Toxoplasma will try to pool, in simple terms, nutrients from mitochondria, that's part of why they're trying to, get to the mitochondria. Do you think that some therapeutics for inhibiting toxo down the road, especially in a chronic capacity, could try to limit its ability to pool those nutrients from mitochondria?
It's a very interesting point. I think at the acute phase, this is feasible because we've shown that if we add molecules or pharmacological agents that promote the ability of mitochondria to take up fatty acids, the parasite grow more slowly. However, the chronic setting, I think, is still a bit of a mystery. We don't exactly know what the nutrient needs are for successful chronic infection. So I think we need to understand those needs first before we can delve into what's the most optimal way to modulate host cell metabolism to decrease the growth of the chronic stage of the parasite.
That makes a lot of sense. I do think there is a tendency to take what pathogens can do in an acute state and then project that onto what they can do in a chronic state, when sometimes it can be so different because over time, the pathogen changes so much of the way it behaves as it becomes more chronic. So
exactly. And the way it interacts with its host cell.
Totally. Yeah. Cool. Yes. So, well, then you're you're studying whether toxo may reactivate in patients with COVID, and you're continuing to work on these metabolic interactions with toxo. I have a question with the identifying toxo in, let's say, a COVID patient. Let's say it's in the brain. Can you find its genetic material in the blood, or is that a challenge? Because with a lot of the pathogens that we are, for example, also connect with a similar trend, Epstein-Barr virus or something, one of the issues is that you'll rarely find the genetic material of EBV in the blood. Sometimes it's less likely to be there because it's a neurotrophic pathogen, right? Do you face the same challenge with toxo, or do you think that enough gets into the blood that you can find the genetic material of it? Or are you not sure?
I'm not sure. It's an important question. We don't know. We hope that it would be present either in the blood or as present at serology level. So that if there was a reactivation in the brain, we should still see this manifest in an increased abundance of IgGs that target Toxoplasma. So we don't know. And I presume this has to do also with how much it's reactivated in the brain.
I would agree exactly. If there's major reactivation and genetic material somehow starts to shed or something, maybe, but it depends. But what you're saying is the antibody responses are more likely to be able to be identified in blood. What about epitope? Does Toxoplasma create any proteins or epitopes that you could look for, or that you do?
Yeah, definitely. I think this is how. So there's different serology tests to toxo. Most of them are not toward the actual proteins that the parasite secretes, but the proteins that cover the parasite. And that allows us to know just whether or not there's the presence of the parasite is being detected by the immune system. As if the infection progresses or if the infection reactivates, we would see changes in the antibody response to the parasite. So it doesn't necessarily have to be towards the protein that the parasite secretes, but to the parasite in general.
Okay, that makes sense.
Detectable.
Cool.
And we're lucky that because, so what I had mentioned before is that one of the, I mentioned in an immunocompromised patient, Toxoplasma can be a big issue. Another type of immunocompromised patient is a fetus. So if a pregnant woman transmits toxo to her fetus, this can be quite dangerous because a fetus doesn't have a developed immune response. And usually if a patient is infected in the 3rd trimester, it's less likely to manifest in or it's less likely to cause damage in the fetus. And if a patient is infected in the 1st trimester, it's much more likely to have a negative consequence on the fetus. So it's important to understand at what stage a pregnant woman was infected. Was she infected before she even got the parasites, or was it, before she became pregnant, or did she get the infection during pregnancy? And I say this because since there's such an important need for addressing when a patient was infected, or at least a pregnant patient was infected, there's really nice tests that have been developed by the Jack Remington, Dr. Jose Montoya, that enable a clinic to identify how old is this infection. So based on the different antigen profiles, based on the different antibody or the serology tests, does this seem to be an infection within 3 to 6 months or older than 6 months? So I think at least toxo there's nice tools to address that in a COVID setting. How long has this infection been active? When did it reactivate, et cetera?
That's great. Yes, that's great that that's been developed. And I'm also just comforted to think that that testing could be done on a pregnant woman. How often is that testing performed, though? Is a woman who's pregnant, let's say my friend is pregnant tomorrow, will they do any toxo testing on her?
It's a great question. In Europe, it's actually mandated, or at least I should say Europe. And because I'm in Germany, I say Europe. In Germany, at least, and I think also in France, toxo testing is required. I'm not sure how regularly, but at least I think at the onset of pregnancy and at some point during. From what I recall, in France, it's also every trimester, but I could be wrong on whether or not it's required every trimester. In the States, I don't think this is routine. And I think there's a push to make it routine because it can have dramatic consequences. And another example in which it can have dramatic consequences, it's not really routinely tested, is in a transplant scenario. So in a transplant scenario, an organ from a donor is given to a patient that is on immunosuppressive therapy, usually, so that they can accept the organ. If the donor had toxo, you can imagine this can have devastating consequences for the recipient. But it's not routinely tested. So I think in some cases it's really important to understand to be able to test for toxo.
Yes,
and it's not yet.
Absolutely, especially, well, no, in both the organ transplant recipient seems obvious, and then but in the pregnant mothers as well. I'm glad to hear that's happening in Europe, and I do think it should be pushed for here more in the USA. That's really interesting. I didn't know that because yes, I've never had anyone tell me they were not that they necessarily know their blood work, but that they were tested for Toxoplasma or anything like that.
Here it's exactly, it's
interesting.
Normal that they are tested.
Because one of the things I think about with chronic disease is obviously we know that certain conditions or just symptoms sometimes run in families. And a lot of times it's assumed that that is because we inherit human genes. Human genes are passed through our families, but we also pass pathogens through families then. We can pass, for example, toxo could be passed from mother to child. So there could be somewhat of an inherited aspect to disease that involves Toxoplasma.
Absolutely. Then it's transmitted. I guess it's more semantics on whether or not this is inherited. In the end, it is genetic material that's given from mother to fetus, but it can definitely have negative consequences for the fetus. And one signature I think that is more common in certain countries in South America is chorioretinitis, so a fetal infection. But that manifests in scars in the eye. Okay. So even if the fetus is exposed to Toxoplasma in the womb, it might not necessarily get the Toxoplasma. It might just get some of the effects from inflammation or something like that. Well, I don't know if we can say that, but if a woman, just because a woman has an infection doesn't necessarily mean she will pass it on. I think depending on the trimester, you have different probability, but it's never 100%. And hopefully it's not.
I know.
But if it is transmitted to the fetus, it is a high likelihood that there will be detrimental health consequences.
Yeah. Okay. How many labs are studying toxo? It feels like too few.
I think a lot are, well, of course, because
Okay.
I'm a toxo researcher.
Okay, good.
But I think as with several infectious diseases, it's much harder to do these studies in humans. There's such complicated red tape that one has to get through to gain access to patients, or even samples from patients. Or I can tell you here, it's been a nightmare to try and gain access to just some serum samples that are available that are otherwise not used to just test whether or not they have toxo. So it is not easy, but there's I think a very big and lively community of toxo researchers.
Good. I'm glad to hear that. I'm glad you're running in lots of toxo circles, because it just seems, for the number of people infected and what the parasite is capable of driving if your immune system isn't in great shape, it just seems like such an important topic.
Yeah, I mean, I'm glad obviously that you're mentioning this, but I think you're of course more invested in the infectious disease aspect. But it's as with the squeaky wheel gets the oil, right? And so asymptomatic infections, at least that appear asymptomatic, are not going to gain so much attention relative to chronic heart diseases or metabolic diseases or cancer that are much more obvious burden and economic burden. But I do think there is also a question of whether or not chronic diseases or chronic infectious diseases affect any of these other human conditions. Because at the end, toxo does live in cardiac tissue. It is a pathogen. It affects immune signaling in a way. We know some of the metabolic and immune signaling are dysregulated in diabetes, for example. So are there signatures of chronic infectious disease in any of these other conditions? But I guess we won't know until we actually look.
Absolutely. That's what I was going to say too, because for example, Epstein-Barr virus can drive cancers. It doesn't have to, but it can be an oncogenic or cancer driving virus. So you have these connections between pathogens and chronic conditions.
Yeah,
so it seems like Toxoplasma could be one of those where we have conditions, like you said, certain cardiac conditions or diabetes, where it's not like we nailed exactly what's going on there. So there's still an open question of whether these pathogens may be part of the process that contributes to some of the chronic conditions that get all that attention and funding. So I think that they're
absolutely. And then they shine in the light a little bit. For example, Helicobacter pylori, which is of course a bacterial pathogen that lives in the stomach, has been shown to drive, or has been associated with certain gastric cancers. So I think there's going to be a lot more pathogens that do have associations or can potentially drive chronic syndromes that are a much bigger problem.
Yeah, agreed. Really interesting. Well, cool. That's great. Then it's really always interesting to talk to you because I'm particularly fascinated by Toxoplasma. I don't know. I just the parasite, the way it can infect the brain, the way this is understood. It's just such an exciting area, I think, in which if we better understand toxins behavior, exactly like what you're studying, there's a lot to learn there. So keep up the good work.
The remarkable pathogen. Thank you so much for your questions. It was a lot of fun. And if you do have long COVID researchers who are interested in looking at whether or not toxo is making an appearance, please put me in touch with them.
I definitely will start to ask around. Yes. Okay. Yes.
All right. Cool. Thanks so much,
Cool,
Amy.
Lena. All right.
Nice.
Bye.
Bye.
Bye.